I wanted to ask about biomembrane. I mean, what does it do exactly? Can you please explain?
Okay, so what is the biomembrane? It is the kind of border of the cell. So if you think about the cell with the nucleus and all these Golgi, etc., you have to isolate it from the environment, and you do it with this biomembrane. So it's kind of envelops the whole cell. So let's call it so it's the isolation of the cell structures, etc. Why do we need it? Because if we don't have it, nothing holds these contents within the same place in water or wherever. And this makes this cell kind of dead, or it cannot function at all. So the thing that this biomembrane is thought to be the first thing that made a huge step from nature towards live nature. So if we have something to isolate and in this case control the whole processes and functions, then we can talk about the living organisms. So we isolate first of all reactions. And these reactions are the basis of life itself. Also, we isolate the environment, so it's internal environment. We also isolate temperature. And because we can control the temperature, we can also control the chemical reactions and their outcomes, because some reactions with a higher temperature, they can increase or decrease their velocity, which means the efficiency of the reaction is in the dependence of this temperature. So it's also controlled by isolation of these contents from the environment. So this is what the biomembrane, and we have, if you looked at the image, we have the two layers of phospholipids and triglycerides, and we have so we have first hydrophilic part and hydrophobic. This one is also hydrophilic. So these two parts, I mean, the hydrophilic and hydrophobic, really isolates the cell from usual diffusion. Otherwise, it would be sorry, it would be really difficult to at least control water, because if water can go through the first hydrophilic layer, it cannot go here, for example. Sorry. And this is the main function of the biomembrane. Also, we have proteins that make up the channels or receptors.
Excuse me.
Mm-hmm.
So this hydrophobic and hydrophilic phospholipids
are bonded with which type of connection?
It's covalent bond.
Covalent. Oh, okay.
Good afternoon. There is like the most used microscope is the light microscope, stereo. So what is the difference between the stereo microscope and compound microscope? As I understood, is it right? Compound microscope is like several lenses are combined, whereas stereo microscope is the only one lens that allows us to look. No?
No,
Okay.
unfortunately, no, because in the stereo microscope, you have two eyepieces and two objectives. So, just a second. So let's say that you have usual microscope. And we have one objective and one eyepiece here. So whenever we look at something, we obtain a single image or a single representation of the sample. We can have two eyepieces, but one objective. In this case, we still have one image, and we split it onto two eyepieces. So you can also see something like this. And one objective here. So in this case, if you have one objective, you will have only one image of the sample. And in this case, it will be split by the mirror into two rays. So it will go like this. And this is not a stereoscopic one. Stereoscopic means you have two eyepieces and you have one objective per the eyepiece. And of course, it doesn't matter if you have stereoscopic or monoscopic. Both of them might be compound. Compound means that in your objective, for example, you have more than one lens. So the system actually includes a lot of different types of lenses, but you don't need to know which lenses we use. You just need to know that there are more than one piece of lens. And if you go to the eyepiece, usually it's made up of two lenses. So 1 and 2. So this is called compound. It means you have like a very sophisticated structure of the microscope. If you think about the stereoscope, you have to understand that instead of one image, you obtain two images, and your brain actually rebuilds these two images into one 3D structure, and you can actually see which contents, which organelles or which cells are closer to you and which are farther. In the monoscopic one, so you have one objective, you will not be able to distinguish the distance of the sample from you. Is it clear?
Yes, thank you. Can I have one more question? So here about the organelles. So it was written that ribosomes produces peptide chains. That means they are used in the production of proteins. That's right?
Yes. That's
Okay.
right.
So endoplasmic reticulum, especially the rough endoplasmic reticulum, is also used in the synthesis of proteins. So both of them produce proteins. That's right?
It is.
Okay, so thank you.
So the thing is, ribosomes, they can float in the cytosol without attaching to the endoplasmic reticulum, or they can be attached to this endoplasmic reticulum, and the produced proteins will stay in this reticulum. This is the only difference. If you have one in cytoplasm, the ribosome, it will produce so-called local proteins that will be used inside the cell. If you are producing proteins through the endoplasmic reticulum, it will most probably go to the secretion or to the biomembrane. So it will be exported.
So here is written that mitochondria has its own DNA, mitochondrial
Yes.
DNA. So does it have any genomes in it, and what is the difference between the simple DNA and the mitochondrial one?
Okay, good question. Nice. So we have a cell, and its genome is inside the nucleus. And the genome means all the information that is being encoded in the DNA. So DNA means genome. And if you have the mitochondria, you can see that they even have the shape of bacteria. They are very small and prolonged. They look like the bacilla and very close to their sizes too. So if you have these mitochondria, so let's say in this nucleus you have 20,000 genes. And these genes are totally controlling everything inside this cell. If you take a mitochondria, it has only 37 genes. And these genes used to be inherited. They are not used to be. They are inherited only after the division of the mitochondria itself. And the division of mitochondria is not related to the cell cycle. Cell division and cell cycle. So it's not related. If the cell needs more mitochondria, it just gives the signal to this mitochondria and they start dividing. That's all. And these 37 genes, they are controlling the production of internal proteins. And if you take the usual nuclear DNA, it also produces some of the genes for mitochondria only. And this means that previously the mitochondria had much more genes than 37, but they migrated here, and most probably because they were prone to the mutations. So they kind of, let's say, decided to keep 37 genes that are very reluctant to the mutations. So they are rigid and safe. They cannot mutate, okay? If you take the mitochondria from today and from 200,000 years before us, they will have almost the same or the same gene sequences, both of these mitochondria. So it's not the last thing that is special about the mitochondria. They also have their own ribosomes. And the thing is, they have prokaryotic type of the ribosome. They are smaller, and they are also controlled by these 37 genes. And if you give these mitochondrial ribosomes the messenger RNA from the nucleus of the cell, they will not be able to produce anything because they are not specialized for eukaryotic proteins and eukaryotic genes. And vice versa, if you give mitochondrial RNA to usual ribosomes, they will not do anything because they are not matching. So the question was about the difference between nuclear and mitochondrial ones. So the mitochondrial DNA, like this, is very small and is circular. If you take the nuclear DNA, it's huge. So outside these 20,000 genes, we have about 60,000 genes that do not code proteins. So it's huge. It's bound to proteins. And it is isolated from the cytoplasm. While the mitochondrial DNA is floating inside the mitochondria. This is the difference between these two. And this is why we say that at some time in a very distant past, we had these obligatory bacteria that lived with the eukaryotic cell. And at some point they were specialized into energy factories. So maybe in many millions years, we will have fewer genes than we have now in the mitochondria. Okay, so question from Tamaris. Energy store or storage of energy is usually lipids or triglycerides or sugars, and more specifically for animals and for mammals, it is glycogen because it's very packed. It's very efficient in holding this energy between these bonds. So you have, let's say, sugar, and let it be glucose. All of these chains, all of these bonds, they have energy. So this energy is stored within the bonds of the sugar. And by aerobic respiration, we can extract this energy by reducing these glucose to CO2 and water. So of course you need oxygen here. And from one molecule of glucose, we obtain 38 molecules of ATP. We can easily use this energy, unlike the energy that is in glucose. So before we can produce energy from glucose, we need this ATP step. Otherwise, it won't work. And usually, just because we are not always using this much energy, we can also store it for bad time. So let's say we have stress or conditions very close to death. In this case, we will need a lot of energy very quickly. That's why we need to store some for future. And we store it in the glycogen. And this glycogen is a huge chain or huge polymer of these glucose molecules bound to each other with a covalent bond. So it has branching so that it can take as little space as it's possible. And whenever we need it, we add water here and break these bonds between sugars, and then use them in the aerobic respiration. If we want to use, if for example there is no glycogen anymore, we can use triglycerides or other, for example, for proteins. This energy, the energy that is stored within huge chains of fatty acids, it's not as easy to extract as in glucose, for example. There are lipolysis reactions that can do that, but the outcome or output of the energy is not as much as in sugars. And if you take proteins, it first goes to the construction of glucose, and then it goes to this aerobic respiration. That's why when people stop eating, they easily lose weight. So they give kilos because they lose triglycerides. First of all, they lose glycogen, then they start losing the triglycerides, and then proteins. And whenever a person is ill, illness usually is a huge problem for an organism that needs a lot of energy, and sometimes we don't have much air or much oxygen to use. That's why we switch from the aerobic respiration to anaerobic respiration. And this is very inefficient because from one glucose, we obtain only 2 ATP molecules. But whenever the person is ill, we have to go with this pathway too, because let's say there is a pneumonia and a person cannot really supply all tissues with oxygen. And that's why in some illnesses, in some diseases, we really lose a lot of proteins and triglycerides, and we lose weight.
Hello, good evening. My question is about second lecture about chemical bonds. So, in water, hydrogen and oxygen bonded with each other with covalent bond, but their molecules bonded with hydrogen bond.
Hydrogen bonds, yes.
Yes. So, is water like the only thing which can have like 2 bonds combined, or are there like any other things?
Could you please repeat, because I couldn't hear your question?
So question is water like the only thing which can have like 2 bonds combined, or is there any other things?
As I shown in the previous lecture, when I was explaining the DNA, for example, they both have covalent bonds, and because of these covalent bonds, one of the atoms inside this molecule with higher electronegativity, it can actually form these local regions. So just a second again. So let's say that we have one nitrogen inside the molecule, and it was, let's say it's the nucleotide, okay? The part of the nucleotide. So this nitrogen forms these covalent bonds with carbon here. And since it's a very electronegative atom, it kind of tries to pull the orbital or electron cloud to itself from both sides. So electronegativity is with a minus sign. In this case, these guys, they will have slightly positive region of electronegativity. So in this case, this nitrogen can attract one molecule of water. So it forms this hydrogen bond with the water. So this is HOH. Because this hydrogen has plus in its electronegativity. It gives its own electron to oxygen, and this guy too. So plus. So if you take water, in this case we have two regions of plus sign or slightly positive charge, and one region of slightly negative or just negative. That's why this nitrogen, it has slightly negative region and it can attract slightly positive. Is it clear?
Yes, thank you. And another question is, in 47th slide, there is the thing like hydrogen bonds among many water molecules are stronger than an ionic bond between two ions, natrium chloride, I guess. So how that
Sodium
works?
chloride, huh? So the thing is, again, you have a lot of molecules of water.
Mhm.
Let's say, let's show them like this. So one part is plus, another is minus. And you have really a lot of them. And also at this part, you have a crystal of sodium chloride. And in sodium chloride bond, we have ionic bond, and it means that the sodium atom gives its electron to chloride, and it receives it, so it becomes negative, okay? So these guys goes to chlor. That's why this bond formed between these two elements is called ionic. But now we have a lot of these water molecules, and they start getting to sodium or to chloride. In this case, with the negative side. So again, negative side and negative side. And to chloride, it comes with the positive side. So it's plus, plus, and plus. So if you take just one molecule of water and sodium chloride, the ionic bond is stronger than this hydrogen bond. But whenever we have a lot of these molecules of water, they kind of overcome this ionic bond. And that's why this ionic bond is torn, it breaks down, and these two ions become separate. In this case, we have sodium without one electron and chloride with one excess electron. And they are surrounded by these molecules of water that really keep them apart, so they cannot come to each other. That's why in total, so it's the number of H2O molecules. In total, its energy, so energy is higher than one ionic bond. Is it clear?
Yes, so that means like it can also apply to any other ionic bond, yes?
Yes.
Okay,
So
thank you.
that's why we have the dissociation of molecules in water. So we dissolve some matters, some molecules. So let's say you have sugars, and if you try to dissolve them in water, it is possible because of these hydrogen bonds, because they can do the same as shown here. So they tear these ionic bonds, and these sugars, they just enter the solution in water.